My dear students, in the previous class, we had discussed about BT cotton. In today's class, we will talk about the same topic, major topic, that is application of biotechnology in agriculture. In that, the subtopic pest control plants are transgenic plants against pest control plants. In that, we have discussed about one nematode. Nematode acts as a pest. Now, what is a pest here? Pest is a group of animals that damages the plants. Doesn't get confused with insects. Insects are, of course, damages the plants, but there are some beneficial insects, like how we have discussed in the previous classes, like pollinators. In the 9th chapter, we have discussed. What are here pests? Pests include insects also, rodents like rat, mice, bandicots, squirrels, other insects and other animals which overall damages the crops or the plants, the yield, are collectively known as pests. Now in this transgenic plants, the next is pest controlled plants. Before we go into the topic, two small topics I will give you as introduction. One pest is known as Nematode incognita. I will give introduction to this plant. This pest. Then I will give introduction to one more bacteria known as Agrobacterium tumefaciens. Let us try to understand this concept first. Then we will talk about RNA interference. It's a technology. What is Nematode incognita? It is a nematode. What is this? Nematode. What do you mean by nematode? The animal, the worm that belongs to phylum Ascalmanthus, class Nematoda. Earlier it was this phylum was also known as Nematohelminthes. Later it was named as Ascalmanthus. The major class in this is class Nematoda. To that class Nematoda, this worm belongs to. What is a Nematode incognita? It's a nematode. What is a nematode? It's a worm. You got my point? Then, why should we discuss this here is because this worm acts as a pest. This nematode acts as a pest and it damages the tobacco plants and other plants. Where it damages? At the root level. What level? At the root level. Okay? In the root system. Just to understand, I am showing you a very rough diagram over here. Right? Roots, these nematodes will be present in the soil, underground soil, because it infects roots means it's very much understood that it is found present in the soil. Due to the secretion of a protein, these nematodes will get attracted here. Right? These worms will get attracted. They come into the roots, they settle onto the roots of these tobacco plants and other plants. Then they start damaging the roots. How this happens? Nematode incognita is a pest. What kind of pest? It's a nematode. What do you mean by nematode? Nematohelminthes. It belongs to class Nematoda of phylum Ascalmanthus. It's a worm. What is it? Worm. Where do you find present in this? It is present in the sand, in the soil, surrounding the roots. Now, how it infects in a tobacco plant roots? Certain chemicals are secreted, secondary metabolites are secreted, due to which it get attracted towards the roots of the plant. To understand this concept, you have studied about mycorrhiza, biofertilizers, how they get attracted towards the roots of the plants. They are beneficial, whereas this one is a harmful. Right? These worms will be found present in the soil surrounding the root. So here, because of the secretion of that protein, these worms will get attracted to roots and they harm the roots by settling onto the roots. So they damage the roots. Once the roots are damaged, the whole plant will get damaged. Right? So to control this, many pesticides, insecticides, pesticides, chemicals were being sprayed into the soil, right? Were being poured into the soil around these roots, were used. But what happens here? What happens? It causes soil pollution. Moreover, these chemicals are so strong that to kill a worm we have to use so strong that they also damage the roots of this plant, and they also cause soil pollution. So this was not the best solution to overcome this problem. Because of this, to overcome this problem without the chemicals, without pesticides, what scientists thought, what biologists thought, what biotechnologists thought is to overcome this problem from the genetic level. In your molecular biology chapter, you have studied protein synthesis. Fine. So for every synthesis of a protein, one gene is responsible. One protein concept you might have understand. So here, what is the concept here? The protein is synthesized here. Certain metabolites are synthesized. Protein is synthesized here. So what biotechnologists thought in a way that why can't we stop the synthesis of this protein into the soil, surrounding soil, so that these worms should not get attracted. The worms should not get attracted towards the soil. So how to make that protein not to be synthesized in the soil? To stop protein synthesis, you have to go for protein synthesis mechanism, mRNA, right? All those techniques, you know how protein is synthesized. In previous chapter you have studied. So now that they thought of stopping this protein not to be secreted from the root, so that this worm is not getting attracted towards the roots. So this concept was there. To introduce this or to do this, they used one natural genetic engineer. It is known as agrobacterium. Agro refers to soil also, agriculture also. Anything concerned with the agriculture or concerning with the soil is known as agro. Bacterium here refers to rod-shaped bacteria, soil bacteria. Gram-negative bacteria, most probably is rod-shaped gram-negative bacteria. Tumefacial. What is now? Tume refers to tumor. Facial refers to formation. The bacteria that infects the plant and causes tumors into the plants. Such a bacteria is what is known as Agrobacterium tumefaciens. This is also a soil bacteria. What bacteria? Soil bacteria found present in the soil. What is the significance of this bacteria? Means to say that this bacteria normally infects the dicot plants. What plants? Dicot plants. Infect the dicot plants. When it infects the dicot plants around the stem region, clear? These are the root, for example, right? This bacteria is found present in the soil, very close to stem region, just below the soil. It infects. When this bacteria infects the dicot plants, it is a neat point. Do remember that Agrobacterium tumefaciens only infects the dicot plants. It do not infect monocot plants. Like for example, right? Like grass, monocot, it cannot infect because of the lack of vascular bundles here. Fine, all those things. Fine, you have studied the difference between monocot and dicot because of the KMM, all those things. Fine. Anyway, my dad was bringing here some grass and putting, so I thought to show you. Anyway, when it infects dicot plants, what happens here? After the infection of the dicot plant, there is a formation of tumor. What is it? Tumor formation. Hence it is known as tumefaciens, tumor forming bacteria, Agrobacterium tumefaciens. If you understand the background, how the name has been suggested to this bacteria, you will understand the whole concept. Agro, present in the soil, something concerned with the soil, something concerned with the agro or agriculture. Bacteria, bacterium, tumefaciens species. One more bacteria is there, known as rhizogens. Agrobacterium rhizogens, that itself we can understand. Rhizo refers to root here. Fine. In mycorrhiza also same terminology. Rhizo refers to root. Rhizogens that generates, fine, tumors like in case of roots. Whereas this bacteria, Agrobacterium tumefaciens, infects the dicot plants naturally. Normally it infects the dicot plants. When it infects the dicot plants, it forms tumor-like structure near the stem region. Hence, that infection is known as crown gall disease. What disease? Crown gall disease. Crown, this region immediately above the soil where the stem starts, that region is referred as crown, like a ring, like a crown region. Gall, crown gall, swelling, tumor formation. Roots, stem, toward the stem like this, crown gall disease. This can be also asked, name the species or name the organism that causes crown gall disease. Which one? Agrobacterium tumefaciens. Fine, clear. So it infects. So normally, naturally it infects. Now scientists are, hence it is also known as and moreover, now if your doubt comes to your mind, how this tumor is caused, one more very important point, biotechnological point now, fine, to tell you how this tumor is caused, means to say that, fine, in your 11th chapter of biotechnology, you have studied a plasmid. Here, what is plasmid? Extra chromosomal. Yes, what is plasmid? Extra chromosomal, double-stranded, self-replicating, circular bacterial DNA is known as plasmid. This is the complete definition given by this our scientist. What is his name? Timin and Baltimore. Fine. Timinism, reverse transcription. Fine. Baltimore was his co-assistant. They were together. Timinism continued his research. Baltimore, after some research, he stopped research. He started writing books. He has done a basics of biotechnology. Has done a book. Basics of molecular biology also has done. So in that, the complete definition of plasmid is given in your 11th chapter. You might have studied. Extra chromosomal, self-replicating, double-stranded, circular bacterial DNA is known as plasmid. You have studied in 11th chapter, or you will be studying in 11th chapter. So that plasmid is used as a vehicle, as a vector vehicle to transfer genes, to transfer DNA, RNA. Similarly, so this bacterium has a plasmid known as TI plasmid. What plasmid it is? It is known as TI plasmid. What is TI? Tumor inducing plasmid. TI plasmid. So scientists thought that they have this bacteria has this plasmid naturally infects. Through this plasmid it can transfer DNA and RNA. So this bacteria, they named Agrobacterium tumefaciens as very important point. Natural genetic engineer. This can also be asked in your NEET. Identify the natural genetic engineer. Fine. Agrobacterium tumefaciens. These two concepts I wanted to put you in your mind. Next concept, continuation of this. How to overcome this problem by using this bacteria is a combination of that. That technology is known as RNA interference. In my next class, I will deal about that. I hope it's clear. Very simple topics. Malignant incognita, nematode, pest. It infects tobacco plants because it is found present in the soil. Then about this bacteria, right? This bacteria, Agrobacterium tumefaciens, found present in the soil. 13 minutes. Okay. I'll repeat now. Right? Pest control management. Malignant incognita is a nematode. Is a worm belonging to Ascomyces class Nematoda. This worm is found present in the soil. This worm, because of the secretion of a protein from the roots of tobacco plant, this worm gets attracted towards the roots. After attraction, it infects the roots, it damages the roots, causes infection to the root, damages the root. So thereby the plant will get disease, and the product, it affects the product, it affects the yield. Now how to overcome? To overcome this, many pesticides are used, but these pesticides are soil pollutants, right? And they also cause other beneficial microorganisms found present in the soil, which are used as biofertilizers. It also causes soil pollution, it kills the other microflora present in the soil, right? Phylloplane, similarly here, right? Rhizoplane, the microbes present in the root, beneficial microorganisms, right? Then also affect the roots and also kill many useful insects. So to overcome this problem, scientists thought in a biotechnological way, at the generic level how to suppress this gene, how to make this gene non-functional. Which gene? The gene that secretes this protein. Molecular value, one gene, one protein concept. Fine. So one gene is responsible for the synthesis of one protein. So they thought in a genetic way, why can't we alter the plants? That what alteration on the genetic level is known as transgenic plants. They thought that, biotechnologists thought that, why can't we make a pest controlled transgenic plant? So for that, to introduce the DNA or RNA or a gene to suppress the other gene, they thought of using vehicle as agrobacterium tumefaciens. Now what is agrobacterium tumefaciens? Agro, soil, bacteria, tumefaciens, tumor forming bacteria. This bacteria normally naturally found present in the soil, around the soil. It infects these dicot plants, and while it infects the dicot plant, it transfers a plasmid. What is a plasmid? Plasmid is an extra chromosomal, double stranded. What is extra chromosomal? Apart from its own chromosome, there should be one more DNA, extra chromosomal DNA, double stranded, circular, self-replicating. It should not depend on the genomic DNA to replicate. Self-replicating bacterial DNA is what is known as plasmid. So this plasmid, it has a plasmid known as TI plasmid. What is TI stands for? Tumor-inducing plasmid. So whenever this bacteria infects the plants, it transfers the tumor-inducing plasmid. Because of that, tumor formation takes place around the crown region. The disease known as crown gall disease. These two concepts you try to remember. Read the study material, go through the study material, read your NCERT textbooks, fine. I will continue in my next class, next session. These two concepts you repeatedly watch. Keep studying. Do take very good care of yourself. God bless you. Good luck. Take care.